All right, let's give this a try. I think I've got 6 slides on this podcast for you, Alicia. I'm going to try to briefly give you the highlights of mitosis. So before we do mitosis, let's look at this a karyotype of chromosomes. In humans, we have 46 chromosomes. We get one set of 23 chromosomes from our mom and one set of 23 chromosomes from our pop. So if you look at this slide, you can see at the top it says chromosome 1, 2, 3, 4, 5, 6, 7, 8, down to chromosome 22, and then you get an X or a Y. If you get an X and a Y, you are male. If you get an X and an X, you're a female. You probably know that. But the important idea here is to see that these chromosomes are not duplicated, for one thing, and then another thing is these chromosomes are called homologous pairs of chromosomes because if you look at the banding pattern, you can see that they are similar. So you get chromosome 1 from mom, chromosome 1 from pop, chromosome 2 from mom, chromosome 2 from pop. And they're called homologous chromosomes because they have pretty much the same genes on them. So a chromosome then is just one of 46 pieces of DNA in our cells. So like in bacterial cells, their DNA is circular, so they just have one circular piece of DNA. But we have 46 pieces of DNA in our cells. And then a gene is just that region on a chromosome that has directions or codes for a single protein. So this is a cartoon of the cell cycle, which you've probably gone over. You can see that there are 2 major phases on the outer ring of the cartoon. There's interphase, and there's 3 segments within interphase. There's the growth phase, and that's G1, and then S phase is the DNA synthesis phase, and G2 is where the cell is preparing for division, aka mitosis. And then there's the mitotic phase, or the M phase. And within M phase, there's pro, meta, ana, telophase, and then there's a final phase within mitosis called cytokinesis when the cells split apart. And that's what it shows at the very bottom of the cartoon. It shows that the chromosomes are lining up on the metaphase plate, and then on the far left corner, or lower left corner, you can see the cells are starting to split apart to complete mitosis. So in the G1 phase, the cell's just doing its regular job. It's growing, and then if it gets a signal, let's say you are a somatic cell in the intestine, and those cells have to be replaced all the time. So you get a signal that it's time for you to undergo mitosis and make 2 daughter cells. So you'll get that signal, and you'll go through S phase, where you actually replicate your DNA. Sometimes that's called DNA duplication, but it's the same thing, replication and duplication. And you need to do that because remember, you're ultimately going to go to mitosis and make 2 new daughter cells. So G2 phase in the cell cycle, you're just getting ready for mitosis. And then the M phase is when you actually divide the cell into 2 equal sets. So remember, you duplicated your chromosomes in S phase, and now each daughter cell at the end of mitosis is going to get a full set of chromosomes, and that full set would include 46 chromosomes. One set you initially got from your mom, one set you initially got from your pop. Okay. So let's look at mitosis prophase, and sometimes they call it prometaphase. So if you look at the cartoon, you can see that there's 3 words that are really close. They sound alike. There's the centromere, there's the chromatid, and there's a chromosome. So in this cartoon, you can see there's 4 purple strands, so that this cell has 4 chromosomes in it. But you can also see that those chromosomes are duplicated. So when the chromosomes duplicate during S phase in the cell cycle, they become sister chromatids. And the sister chromatids, they're just 2 identical pieces of DNA. They're joined together at the centromere. So when a chromosome has undergone S phase, then you can say that they don't really call it a chromosome anymore. They call it a sister chromatid joined at a centromere. Okay, so the duplicated DNA then gets ready to line up on what's going to be called the metaphase plate, and you're going to want to line up your sister chromatids along the metaphase plate. So the centrioles then are those things in gold in the cartoon. The centrioles are going to use mitotic spindles, which are those yellow lines coming out of the centrioles and touching each other. Those are just proteins, and they're going to use that mitotic spindle to actually pull the cell apart at the end of mitosis. So at the beginning of prophase, you're setting up the conditions so that you can split your single cell into 2 daughter cells. Now on the right hand of the cartoon, it shows that the nuclear envelope has fragmented, and then it shows you this other structure called a kinetochore. A kinetochore is just a region of DNA where the spindle fiber connects to the centromere of the sister chromatid. So then in metaphase, you can see that the sister chromatids have lined up on the metaphase plate, and the spindle fibers are attached to the centromeres of the sister chromatids, and the spindle fibers are also connected to the centriole. And so what you're going to do is you're going to pull the centromeres apart. You're going to pull one sister chromatid to one pole and the other sister chromatid to the other pole. So then in anaphase, you just see that the centromeres of the sister chromatids have broken, and now each sister chromatid is migrating toward a pole of the cell. And then at the end, you can see that the DNA is just a single purple strand, so it's not duplicated anymore. That's just a single, it was a single sister chromatid, but now when it's in a cell by itself, instead of being called a single sister chromatid, it's going to be called a chromosome. So at the end of mitosis telophase and then cytokinesis, you're going to get a new nuclear membrane forming around the chromosomes of each daughter cell. So, and then cytokinesis is when the cytoplasm actually splits. So each new daughter cell gets about the same amount of cytoplasm.